We find ourselves at a really exciting point where we've now gleaned enough knowledge from our prior conversation to understand the full details of the electron transport chain and to recognize what makes oxidative phosphorylation different and unique as compared to substrate level phosphorylation. Remember that we have seen throughout the processes of metabolism, whether it be the Ebden-Meyerhof pathway, even the Ettner-Dudorov pathway, or the transition step, or the TCA cycle, remember all of the NADH and FADH2 that we've seen being produced in those processes. Remember that NADH is ready to take its electrons to the ETC, to drop them off, to be converted from one form of energy into another form of energy. In fact, there's several conversions, right? From the energy held in the bonds of NADH to the energy in the form of the proton motive force, and finally to the energy of ATP. So the first depiction of the electron transport chain that I want to look at is an energetic one that shows us how electrons are energetically transferred downhill and from one carrier that desires them less to a carrier that desires them more. Beginning, of course, with NADH. NADH is the first, if you will, the first deliverer to the electron transport chain. It does so at complex 1 of the ETC. So notice that the first delivery takes place when NADH gives its electrons to complex 1, and this regenerates NAD+, which we know now can take its electron or can take itself back to get more electrons from its original enzyme. So to go back to our analogy of Julia and the pizza delivery, we know that she has way less of a desire for that pizza after that pizza's been smelling up her car as she's delivering it to the frat house than do the guys in the frat house. The guys in the frat house, they're more like complex 1. Complex 1 has a much greater desire for the electrons than NADH does. So the transfer is energetically downhill, and complex 1 easily takes on those electrons. Said in a more technical way, complex 1 has a greater standard reduction potential, a greater desire for electrons. Notice that standard reduction potential is listed on this left-hand axis. The greater the desire for electrons, the lower on this axis the molecule would be. So notice that complex 1 is actually lower in standard, or lower on the axis, higher in standard reduction potential. Notice we're going from negative values of standard reduction potential to positive. So complex 1 has a greater standard reduction potential than NADH does. Likewise, the next complex of the electron transport chain that the electrons eventually go to will have a greater standard reduction potential than does complex 1. And throughout the entire chain, the standard reduction potentials get greater and greater and greater. Said another way, as you go through the electron transport chain, the members of the chain have more and more and more and more desire for the electrons. Now, clearly, the electrons that enter as FADH2, they enter at a different place than the electrons that enter as NADH. And remember, Sammy and I had this conversation the other day when she asked about what the difference is. Well, the difference is that FADH2 electrons, they enter at complex 2 instead of at complex 1. Notice that complex 2 has a more positive standard reduction potential than does complex 1. Therefore, what this means is that the electrons effectively enter at a lower free energy state. They have less potential to pump protons than do the electrons coming from NADH. So notice again that FADH2 is a prosthetic group of complex 2. Hey, that means that complex 2 also is known as succinate dehydrogenase, right? Wonder Woman enzyme. So inside of that enzyme, this is where FADH2 is being generated as succinate is oxidized to fumarate in the TCA cycle. Now here's where, when we make that analogy to the ETC being like a highway where there are multiple entry points onto that highway, here's where the entry point from NADH and the entry point from FADH2 come together. They come together at Q. Q is the common carrier. So Q, when it is reduced with electrons either deriving originally from NADH or deriving originally from FADH2 at complex 2, when it takes on those electrons, it becomes reduced, it forms QH2, and once it is reduced, it delivers its electrons to complex 3. Please notice that complex 3 has a much greater desire for electrons than Q does. And likewise, Q has a much greater desire for electrons than complex 1 does, or than complex 2 does. So all the while, we're getting the transfer of electrons from things that desire them less to things that desire them more. And that's resulting in free energy release. The next carrier of electrons in this chain is cytochrome C. Cytochrome C is going to take on electrons from complex 3, and cytochrome C is going to transfer those electrons to complex 4. Complex 4 has a greater desire for electrons than did either complex 3 or complex 1 or complex 2. In fact, it has the greatest desire of all the complexes. Cytochrome C is just a transient carrier, a mobile carrier that can flit around as a peripheral membrane protein from complex 3 to complex 4. Complex 4 is actually the binding site of the molecule with the greatest desire for electrons, the final electron acceptor, oxygen, diatomic oxygen gas. It's going to take on those electrons and it is going to be reduced to water. So as electrons are transferred through this entire electron transport chain, energy is released. We know that that energy takes the form of the proton motive force. Clearly, this diagram is just an energetic one. And we need to take a moment now to look at a much more physical depiction of the ETC. So that prior graph showed us the energetics, the standard reduction potential comparisons, and the energy released in the ETC. But this picture is much more of an actual picture, right? It actually shows us the physical depiction of what the mitochondrial ETC will look like. And this is important to take note of, right? This is the ETC in the mitochondrion. This is because this is the most well-typified ETC. This is for obvious reasons, because of course humans have mitochondria, and we tend to study things that interest us in that way. But also because it is so well-typified, we can use it to make generalizations to any electron transport chain, whether it be prokaryotic or eukaryotic in nature. So let's look first at the NADH electrons as they're getting delivered to complex 1. And with your newly found knowledge of how to name enzymes, you can very easily name this enzyme because you know that it extracts electrons off of NADH, so it is NADH dehydrogenase. So it's a 2-electron transfer from NADH. It drops off both of its electrons at complex 1. Those 2 electrons get passed throughout complex 1, and in that process, the energy that is harnessed from that is utilized to pump protons. And complex 1 is, in fact, a proton pumping member of the ETC, and in fact, it pumps, as according to most recent estimates, it pumps 4 protons for every pair of electrons delivered from the NADH. So this helps, I hope, Sammy, to further explain the answer to your question that you had earlier. Because complex 1 is the site of entry for NADH electrons, and complex 1 pumps 4 protons, there are 4 protons more pumped for an NADH than there would be for an FADH2. Because at complex 2, that's where FADH2 is formed. This is the site of oxidation of succinate. And if you want to, right, draw the TCA taking place inside of the cytosol, or the inside of the matrix of the mitochondrion, draw the succinate going to fumarate and all the other steps of the TCA. And we know that complex 2 Wonder Woman enzyme is catalyzing step 6 of the TCA cycle at the same time as it is being complex 2 of the ETC. So we know that it's double timing there, but what it doesn't do is with the electron transfers to FADH2 and then finally to Q, it doesn't pump protons. So there isn't a pumping of any protons at the site of entry of FADH2. Instead, from both complex 1 and complex 2, electrons get transferred onto coenzyme Q. Ubiquinone is the other name for that, and it becomes reduced. Notice that it is mobile. It's a flitty little guy that goes in the phospholipid bilayer. It obviously is a highly hydrophobic molecule that doesn't mind hanging out in those phospholipid tails. And it does so and makes its way to complex 3. Complex 3 is a proton pumping member, so whether electrons came from FADH2 or whether they came from NADH, they will pump 4 protons at complex 3. 4 protons get pumped there at complex 3, which is also known as the cytochrome BC1 complex, and throughout the transfers, the ending transfer from complex 3 is to cytochrome C. Now, cytochrome C is an epically famous protein. It's a peripheral membrane protein that has been sequenced to look at evolutionary relatedness. It's a simple molecule to sequence, and so it's commonly used and commonly compared. But cytochrome C has its job as moving electrons from complex 3 to complex 4. And at complex 4, we see the more electron transfers in this cytochrome C oxidase complex, and this leads to the pumping of more protons. We know that oxygen binds at complex 4, where it then gets reduced to form water. And here we have it, the final component of aerobic respiration. All along, we've been seeing these thoroughly worn out, totally used up CO2s giving off. And here, finally, we see a water molecule being given off when electrons are transferred to oxygen. So, right, the last of those components given off in aerobic respiration. We also see now that we've established a proton motive force. That proton motive force can be tapped into to make ATP. But here is where we finally meet the machine that does it. This machine is called ATP synthase. ATP synthase taps into those protons to that PMF, and it allows those protons to swoosh down their gradient, and at the same time they're swooshing down their gradient, it turns a turbine. This turbine is the certain subunits called the C subunits of this machine called ATP synthase. As the protons swoosh in, the energy in the form of the PMF is converted to energy in the form of mechanical rotation. This mechanically rotates, and it rotates a subunit in the protein called the gamma subunit, and as this rotates inside of these catalytic subunits called the alpha and beta subunits, ADP gets exchanged for ATP, and you got it. This, my friends, is oxphos. So here we see oxphos taking place in its full glory, the use of the proton motive force to make ATP. That is oxidative phosphorylation. Now, why does that term make sense? Well, it makes sense because all of the energy required to make this proton motive force, which is then tapped into to make the ATP, came from the oxidation of reducing power, NADH, FADH2. So this really is the coupling of oxidation to phosphorylation, ox-phos, right? And that's really what's happening in the ETC. It took Peter Mitchell, who was a scientist that studied this, it took him many years to be able to show that it was in fact the proton motive force that was the source of energy for the phosphorylation of ADP. This was a long coming process, and it wasn't actually until the 60s that he did much of his work, and later that this chemiosmotic hypothesis gained clout. So chemiosmotic hypothesis, a proton concentration gradient, PMF, serves as the energy reservoir for driving ATP formation, oxphos. So we need to do a couple of important conversion factors from this, recognizing that from one NADH molecule, when it is ripped and stripped of its high energy electrons, that the 4 protons pumped in complex 1 result, and of course then these 2 electrons get transferred to complex 3, and 4 more protons are pumped, and then they get transferred to complex 4, and 2 more protons are pumped. So in total, if we were to add up all the protons that get pumped from one NADH molecule, it's 4 plus 4 plus 2, for a total of 8 plus 2 is 10, right? 10 protons in total pumped from an NADH. So we've got a sweet conversion factor that we should write down here. There are 10 protons pumped for every pair of electrons from an NADH. This is such an important conversion factor. You'll also notice we could add something else to that. From every 10 protons that swoosh into the ATP synthase machine, 3 ATPs are made, right? So we also recognize 3 ATPs for every 1 NADH. We could jot that down as well. Sweet, we've got some awesome conversion factors that will come in very useful. We could also do this for FADH2. Recognize, of course, that its electrons do not get contributed to the ETC until complex 2, meaning that they then go to Q, which goes to complex 3, so they pump 4 plus 2, or 6 protons. So another conversion factor, 6 protons pumped per every FADH2, or we could even say per QH2. So we might then estimate how many ATPs could get made from that amount of protons. And in fact, we generally tend to estimate 2 ATP for every 1 FADH2. And as we begin our coverage when I get home, and I'm already looking forward to seeing all of you upon my return, then we will be looking at how we can use these conversion factors to sum up the amount of total energy that could be generated when we consider complete oxidation of glucose in an aerobically respiring cell that has a mitochondrion. So you can all look forward to that and maybe even practice and get jazzed for when I get back.